Good afternoon, everyone. Today I'll be presenting my seminar on the neuropsychiatric aspects of heavy metal poisoning. I'm Dr. Riya Gupta, junior resident in the Department of Psychiatry. So, in the following seminar, we will be covering about the introduction, the characteristics of heavy metal poisoning, the global burden, the sources of exposure, symptoms, the mechanism of neurotoxicity, the neuropsychiatric symptoms of the specific heavy metals, the treatment, and the summary. And the end, I'll be citing about the references. So heavy metals are basically the elements with high atomic weight and a density of more than 5 gram per cubic centimeters. These metals are toxic at low concentrations and can accumulate in biological systems, leading to adverse health effects. The common heavy metals for which poisoning can occur are lead, mercury, arsenic, cadmium, and chromium. So, coming about a little bit of characteristics of heavy metal in human body are first is the toxicity. Even small amounts can be harmful. The next is bioaccumulation. They tend to accumulate in the body over time, especially in tissues like brain, liver, kidneys, and bones. Persistence. Heavy metals do not degrade over time, which means that once they enter the body or the environment, they remain a long-term threat. Coming to the global burden of heavy metal poisoning. According to WHO, the lead exposure contributes to an estimate of 0.6 million deaths annually due to cardiovascular and kidney diseases and neurodevelopmental effects. Mercury poisoning, especially from contaminated fish, has led to significant health issues in the region near industrial zones and coastal areas. This is a picture which tells us about the density of the heavy metal poisoning, which, as we can see, is pretty high in India. So, these are certain sources like mercury is seen in fish and sushi, lead in broth and wine, arsenic and mercury in supplements, cosmetics, and rice. Lead is also present in unfiltered water, cadmium and heavy metals in cigarettes and e-cigarettes. Coming to sources of exposure, we can have either environmental sources, dietary sources, and occupational sources. For environmental sources, industrial pollution, especially emissions from factories, power plants, and mining activities. Heavy metals like arsenic in drinking water, particularly in areas with industrial runoff. Mercury and lead in air from fossil fuel burning and waste incineration. Soil contamination, heavy metal can enter the soil through agricultural practices like pesticide use or by industrial waste. Dietary sources. Contaminated food. Fish and shellfish can contain high levels of mercury, while grains and vegetables can absorb lead or cadmium from polluted soil. Toxic fish. Some species, like tuna and the swordfish, accumulate high levels of mercury, putting humans at risk when consumed. Occupational exposure. Workers in certain industries like mining, construction, battery manufacturing, etc., may be exposed to heavy metals such as cadmium, lead, and arsenic. Healthcare professions working with mercury in medical instruments like thermometer, dental amalgam may also be at high risk of exposure. Household and consumer products, lead is found in old paint, especially at homes. So these are basically the routes how we can get the poison into how we can get the heavy metal into our body by inhalation, double contact, and ingestion. So routes of exposure include ingestion. Heavy metals can enter the body through contaminated food and water. Inhalation, breathing in dust, vapors or fumes from polluted air or occupational environments like inhaling mercury vapor. Dermal contact, exposure can also occur through skin contact with contaminated surfaces or products. Example, lead-based paints or arsenic in wood preservatives. Coming to symptoms, heavy metals can lead to a wide range of neuropsychiatric symptoms such as cognitive decline, which includes memory loss and learning difficulties, mood disorders like depression, anxiety, irritability, psychosis like hallucinations and delusions, motor dysfunction include tremors and lack of coordination, developmental delay is common in children which lower the IQ and causes learning disabilities. So, heavy metal poisoning, the symptoms include feeling weak, nausea or vomiting, a scratchy feeling in your throat, numbness or pricky sensation in your hands and feet. This slide comprises of almost all the symptoms for generalized symptoms for heavy metal toxicity. We can have chronic fatigue, weight loss resistance, anxious and irritable, cold hands and feet, metallic taste in mouth, muscle tremors, chronic pain, allergies, insomnia, depression, skin problems, feeling disoriented, digestive problems, headache, brain fog, night sweats, cognitive dysfunction, and poor immunity. Coming to the mechanism of neurotoxicity, it has the following mechanisms. First is disruption of iron hemostasis. Next is oxidative stress and generation of free radical oxygen species, inflammation and immune system activation, neurotransmitter system disruption, alternation in synaptic plasticity and learning, protein aggregation and cellular damage, and gene expression and epigenetic changes. We will be looking at them one by one in description. So this is a diagram which shows that how the toxic metal will cause changes in the synaptic transmission. So first is disruption of iron homeostasis. Coming to calcium iron dysregulation, heavy metals such as lead and mercury interfere with calcium signaling in neurons. Cadmium ions play a crucial role in neurotransmitter release, synaptic plasticity, and cellular signaling. When heavy metals disrupt calcium channels or mimic calcium, they can lead to first is excessive calcium intake influx. Overactivation of calcium channels can result in intracellular calcium overload, leading to the activation of enzymes such as phospholipase A2, protein kinases, and endonucleases. This triggers cell damage, apoptosis, and necrosis. Calcium dependent signaling. The overactivation of calcium signaling pathways can also impair synaptic functioning and reduce neuroplasticity, leading to cognitive decline and memory impairment. Coming to sodium and potassium imbalance. Some heavy metals, particularly mercury, disrupts the balance of sodium and potassium ions across the neuronal membranes. This leads to altered neuronal excitability, depolarization, and disturbances in action potential generation, contributing to again cognitive and motor dysfunction. The next mechanism is oxidative stress and reactive oxygen species generation. Mitochondrial dysfunction. Heavy metals induce oxidative stress by damaging mitochondria, which are the crucial for energy production. Mitochondrial dysfunction leads to generation of reactive oxygen species and reactive nitrogen species, which can damage cellular components including lipid, protein, DNA. For example, the mercury disrupts the electron transport chain in the mitochondria, which results in the production of reactive oxygen species. The impact on the neurons. Neurons are highly vulnerable to oxidative stress due to their high metabolic rate and limited antioxidant release. ROS damages the neuronal cell membrane by lipid peroxidation, disrupt protein function, which causes oxidative modifications and cause DNA damage, basically mutations and strand breaks. This cascade of damage can lead to neurodegeneration, cognitive impairment, and neuropsychiatric disorders. Inflammation and immune system activation. There is microglia activation. Heavy metals can activate microglia, the resident immune cells of the CNS. The microglia, when activated by toxins like lead or mercury, they release pro-inflammatory cytokines like TNF alpha and interleukin 1 beta, chemokines, and reactive oxygen species. Chronic activation of microglia can contribute to neuroinflammation, which exacerbates neuronal injury and impairs synaptic functioning. Coming to the blood-brain barrier disruption. Inflammation and oxidative stress induced by heavy metals can lead to the breakdown of blood-brain barrier. When the blood-brain barrier is compromised, it allows toxic substances, inflammatory mediators, and immune cells to infiltrate the brain, further contributing to the neurotoxicity. Neurotransmitter system disruption. Coming to the dopaminergic system. Heavy metals such as lead and mercury can alter dopamine signaling, a key neurotransmitter which is involved in mood regulation, attention, and motor control. Disruption of dopaminergic pathway is linked to mood disorders, cognitive dysfunction, and conditions such as Parkinson's. Lead, for example, inhibits the release of dopamine and disrupts dopamine receptor activity, leading to motor dysfunction and cognitive impairment. Glutamate and the GABA systems. Glutamate is the primary excitatory neurotransmitter in the brain, while GABA is the primary inhibitory neurotransmitter. Heavy metals can disturb the balance between excitatory and inhibitory neurotransmission, leading to excitotoxicity, that is, excessive stimulation of the neurons, and neurodegeneration. Like mercury, increases glutamate release, contributing to excitotoxicity and neuronal injury, whereas cadmium and lead can also disrupt the GABAergic signaling, which can impair mood regulation and lead to anxiety and depression. Alternation in synaptic plasticity and learning. Synaptic function and plasticity. Heavy metals impair synaptic plasticity. This is due to dysregulation of key proteins which are involved in synaptic signaling and plasticity, such as NMDA receptors, which is involved in glutamate signaling, calcium-dependent signaling pathways, and neurotropins like BDNF, that is brain-derived neurotrophic factor. Memory and learning. The disruption of synaptic plasticity contributes to cognitive decline and learning difficulties. For example, lead exposure, especially in children, has been linked to lower IQ, memory deficits, and learning disability due to its impact on synaptic plasticity in brain regions like the hippocampus and the prefrontal cortex. Coming to protein aggregation and cellular damage, amyloid and the tau protein abnormalities. Chronic exposure to heavy metals like mercury and arsenic has been linked to the formation of abnormal protein aggregates such as the amyloid plaques and the tau tangles, which are the hallmark features for neurodegenerative diseases like the Alzheimer's disease. Proteasome dysfunction. Heavy metals can interfere with the proteasome system, which is responsible for degrading damaged or misfolded proteins. When this system fails, the damaged proteins accumulate, leading to cellular stress and apoptosis. For example, cadmium has been shown to inhibit the function of proteasome, causing the buildup of damaged proteins in the neurons. The next mechanism is gene expression and epigenetic changes. Alteration in the gene expression. Heavy metals can influence gene expression by modifying the activity of transcription factors or by directly binding to the DNA. This can result in the activation of genes involved in inflammation, oxidative stress, and apoptosis, leading to neurodegeneration. Epigenetic modifications. Heavy metals can also induce epigenetic changes such as DNA methylation and histone modification, which affect gene expression without altering the underlying DNA sequence. These modifications can lead to long-term changes in brain function and behavior. For instance, lead exposure has been shown to cause epigenetic changes that influence the expression of genes involved in neurodevelopment. Molecular pathways, MAPK pathway, which is the mitogen activated protein kinase signaling pathway. This pathway regulates cell growth, survival, and stress response and is often activated by heavy metals. This pathway it plays a critical role in inflammation, neuronal survival, and synaptic plasticity. Excessive activation of this pathway can lead to neuroinflammation and neurodegeneration. NF-kappa B pathway. The nuclear factor kappa B pathway regulates the expression of inflammatory genes. Heavy metals, particularly arsenic, mercury, can activate the NF-kappa B, leading to chronic inflammation in the brain that contributes to neurodegeneration. Coming to the neuropsychiatric aspects of specific metals, I have covered 4 metals, that is lead, cadmium, arsenic, and mercury. Coming to neuropsychiatric aspects of lead, they are more or less similar only. Cognitive impairment, there is decreased IQ, memory deficits, and difficulty with learning and attention, especially in children. Behavioral changes, increased aggression, impulsivity, hyperactivity, conduct disorder, particularly again in children, emotional disturbances such as depression, anxiety, and personality changes such as irritability and mood swings in both children and adults, and neurodegeneration. Long-term exposure to lead can impair brain development in children and accelerate neurodegenerative diseases like dementia in adults. There is motor dysfunction such as tremors, weakness, and coordination problems called as ataxia, which affect both children and adults. Severe psychological effects. In rare cases, high levels of lead exposure can also lead to psychosis, delirium, and hallucinations. Fetal impact. Pregnant women with lead exposure may give birth to children with developmental delays, lower IQ, and behavioral issues. Coming to the neuropsychiatric aspects of mercury. Cognitive impairment, memory deficits, learning disabilities, attention problems in children and prolonged exposure. Behavioral changes somewhat similar, increased irritability, anxiety, depression, mood swings. Chronic exposure can lead to personality changes such as withdrawal and apathy, motor and coordination issues, tremors, muscle weakness, ataxia, gait disturbances. Neurodegenerative effects include cognitive decline and potential development of Parkinsonian symptoms such as tremors and rigidity. Psychiatric symptoms can cause psychosis, delirium, cognitive dissonance, which includes conflicting thoughts and emotions. Fetal and developmental impact. In the pregnant woman, mercury exposure can result in neurodevelopmental delays, learning disabilities, behavioral issues in the offspring. Coming to the neuropsychiatric aspects of arsenic. Cognitive impairment, which causes memory deficits, learning disability, and a general decline in cognitive function. Behavioral changes, again, mood disturbances like irritability, anxiety, depression. There is increased impulsivity and aggression. Motor dysfunction includes peripheral neuropathy, numbness, tingling, and muscle weakness in limb, which affects the coordination and the motor function, and tremors and ataxia. Psychiatric symptoms include occurs with chronic exposure, which includes depression, anxiety, insomnia. Severe cases include psychosis and delirium. Sleep disturbances. Exposure to arsenic is linked to insomnia and disturbed sleep patterns, which further contribute to mood and cognitive disturbances. The last, neuropsychiatric aspects of cadmium. Again, cognitive impairment like memory deficits, impaired learning, reduced cognitive functions, particularly in older adults. Behavioral changes include mood disturbances like irritability, anxiety, depression, increased levels of stress and fatigue, motor dysfunction. Cadmium contributes to muscle weakness, coordination problem, leading to tremors and difficulty with motor tasks. Peripheral neuropathy, which causes sensations like numbness and tingling in the extremities. Psychiatric symptoms, anxiety, sleep, and mood disorders. Severe cases may again cause psychosis and delirium. Coming to the treatment aspects of the heavy metal poisoning. First, we have to focus on the removal of the exposure, removal from exposure. The first point on this is identify and eliminate the source. The first step in treating heavy metal poisoning is to identify and eliminate the source of exposure. This could involve stopping occupational exposure, example in industries where metals are used or mined, eliminating the contaminated water like installing filters to remove heavy metals, removing lead-based paints in home, particularly for children's environment, changing the diet. We can avoid mercury contaminated fish, for example, or reducing the exposure to contaminated food sources. Supporting care, hydration. Ensure that the patient remains well hydrated as proper hydration helps kidneys to excrete toxins more effectively. Nutrition, providing a balanced diet can support the body's detoxification process. For example, iron supplementation may help to reduce the lead absorption as iron deficiency can increase the lead uptake in the body. Cadmium and magnesium supplementation may reduce the absorption. Calcium and magnesium supplementation may reduce the absorption of cadmium and lead, protecting the bone health. We also have oxygen therapy. In cases of mercury or carbon monoxide poisoning, patients may need supplemental oxygen to support the respiratory function. The next is the chelation therapy. Chelation therapy, it is the administration of a chelating agent that binds to the heavy metal forming a complex that can be excreted in urine. This is a critical treatment for many forms of heavy metal poisoning. The common chelating agents include, for example, for lead poisoning, we can use uh, editate calcium disodium. It is administered IV or IM. This is the most commonly used chelating agent for moderate to severe lead poisoning. We have Succimer, which is DMSA. It is an oral chelating agent for children with moderate lead poisoning with blood levels of less than 45 microgram per DL. It can also be used for milder cases. And we have dimercaprol, which is BAL, often used in combination with other chelators, especially in severe cases. For mercury, we can use dimercaprol, BAL, the primary chelating agent for acute mercury poisoning. We have penicillamine, used for chronic mercury exposure. We have Succimer, an oral chelating agent used for less severe mercury poisoning, often in children. For arsenic poisoning, we have again dimercaprol, BAL, used for acute arsenic poisoning and often combined with other agents like DMSA, Succimer, oral chelating agent, which is used both in acute and chronic arsenic poisoning, particularly in children. For cadmium, there is no widely recommended chelating agent specifically for cadmium. However, certain agents like penicillamine and BAL, which is the British anti-leucite, have been studied with varying effectiveness. Chelation is generally not as effective as cadmium as for other metals. For other metals like iron and copper, For other metals like iron and copper, desferoxamine used in cases of iron poisoning. Example, children who accidentally ingest iron tablets, and trientine and penicillamine used for copper poisoning. For example, Wilson's disease. Coming to the next step, it is the symptomatic support and treatment. For example, if someone presents with you for seizure, in cases of severe poisoning like lead encephalopathy, anti-seizure medications Anti-seizure medications such as benzodiazepines or barbiturates may be required. Dialysis in severe cases of mercury or arsenic poisoning where chelation therapy is ineffective or not feasible. Hemodialysis may be used to accelerate the examination of toxins from the body. We can also provide psychiatric support for chronic exposure, especially in cases of neuropsychiatric symptoms. Psychiatric treatment may be required. This can include antidepressants, anti-anxiety medications, and cognitive therapy. Coming to considerations in special population, children are particularly vulnerable to heavy metal toxicity. Chelation therapy is often used earlier in children with high blood lead levels as they are more susceptible to cognitive and developmental impairments. Pregnant women, chelation therapy is generally avoided during pregnancy unless the blood levels are severely elevated, for example, more than 45 microgram per DL, as some chelating agents can cross the placenta and affect the fetus. In pregnant women, the main goal is to eliminate the source and monitor the patient closely. The next step is follow-up and monitoring. Regular blood testing after chelation therapy, blood levels of the toxic metal should be monitored to ensure that they are decreasing. Regular follow-up is essential to determine the effectiveness of the treatment. Neurodevelopmental monitoring in children, cognitive and behavioral assessments should be conducted regularly as heavy metals can have long-term effects on brain development and function even after the exposure is eliminated. Summarizing everything, lead causes cognitive impairments, behavioral issues, mood disorders, which is commonly also seen in mercury, arsenic, and cadmium. There are various mechanisms for this for the neuropsychiatric symptoms. such as disruption of the neurotransmitters, oxidative stress, and its effect on the calcium signaling. Treatment mainly includes remove exposure, chelation therapy, and supportive care. Then I've stated my references. Thank you. Thank you.